Thanks, Ben, and thank you so much for having me back. I'm actually really excited about this talk because I gave a talk a few months ago. Actually, it may have been like a year now. It just feels like a few months, but I gave a talk not long ago about the first half of this project. So it's fun to actually give like the full circle picture of what we were doing with the Aptamers before. But the title of my talk is Modular Catalysis, Aptamer Enhancement of Enzyme Kinetics in a Nanoparticle Reactor. That is a very scary title, and I hope to break that title down bit by bit throughout my talk. And so to start, because we do say aptamer here in the title, I want to kind of break down what functional nucleic acids are. So of course we know our basic nucleic acids, DNA and RNA, and sometimes we get a little more specific, right? So we know DNA stores our genetic information. Humans are made of DNA, and we know that mRNA. Many people just know it kind of as the mRNA vaccine, but mRNA encodes our proteins, and then we have tRNA and RNA as well. But we get a little bit deeper in the sciences as well, and we have functional nucleic acids, which are nucleic acids that can do more than just their typical, I guess, their biological functions that we know them to be. Specifically, we have aptamers, and aptamers are simply single-stranded nucleic acids. This can be DNA or RNA that wants to bind a specific target. So for example, if we have an aptamer for caffeine, and caffeine is this target here in gray, and I'm actually going to use my laser pointer. This target here in gray is going to want to specifically bind that caffeine, but if you throw something else in there, let's say cocaine, which since I'll be talking about that, this caffeine aptamer won't bind cocaine, so it will stay in this form here. And so again, aptamers are just specifically small, single-stranded DNA or RNA that are capable of binding a specific target. And this will be, of course, that aptamer portion in my title. We also have other fun nucleic acids as well, functional nucleic acids. We have ribozymes and DNAzyme. And if you listen to those two keywords, you have the ribo and then the zyme from enzyme. And these are basically going to be catalytic RNA or DNA. So in other words, you just have now RNA or DNA that can form some type of catalytic function. And then lastly, my favorite that I have never been able to work with just yet, but I really hope to work with this in my career, we have aptazymes. This is now whenever you combine an aptamer with a ribozyme to give you a binding moiety that then leads to some type of catalytic function. And so I want to bring my title back really quickly. So I say Modular Catalysis, Aptamer, Enhancement of Enzyme Kinetics in a Nanoparticle Reactor. So I just explained what aptamers are. But now let's talk about the enzyme portion, because this is going to be kind of the basis of the entire project. And when I thought about this talk for my defense, I thought, okay, well, how can I reach my audience that's not science? And I guess in this case, how can I reach my audience that's not technically biochemists or chemical biologists like myself? And the way I thought about it was to bring about enzymes in medicine and bioremediation, because this is really our everyday lives, and this is really what we know the most, whether you're a scientist or not. And the first thing I thought about was in therapeutics. We typically have enzyme deficiencies in the world, right? And we want to ideally replace those enzymes with enzyme replacement therapy. So if you think about lactate, right? Many people are lactose intolerant, and with lactate, you can simply just deliver the enzyme lactase that is capable of degrading lactose for you. So if you don't produce enough lactase, guess what? You can just take it and replace that enzyme in your body. Next, we see enzymes a lot in commercial uses. I was talking to Ben before this, and he wanted to go make sure his dogs were asleep. And I told him that my dog is actually snoring right now. And when my dog was a puppy, Nature's Miracle was something that really, really saved my sanity in grad school, because it is a safe enzyme-based solution that is capable of degrading pet waste, right? So when my dog would urinate on the rug, I can spray this enzyme-based spray. Because it's enzymatic, it is relatively safe for him, it's safe for me, and it's also safe for the environment. And so again, enzymes show up a lot in our daily lives and therapeutics, but also in our bioremediation. And lastly, I would be doing a great injustice if I did not bring in cosmetics. Self-care is everything in grad school. Self-care is everything in life. And one thing I like to do is get facials. And I'll never forget my first facial. I was getting an enzyme mask on my face. And it was different. I had never thought of an enzyme being used in cosmetics, but enzymes are also capable of degrading dead skin on your face. And so, as you can imagine, increasing efficiency and stability of these enzymes are important, right? Because if they're going to be in the environment, they're going to be things that are going to want to degrade them. If they're going to be in your body's going to want to naturally degrade it if it is not an endogenous protein or enzyme. And then in cosmetics, same thing, right? The environment wants to kind of get rid of these enzymes naturally. And so how do we do that? So typically, whenever you have an enzyme, you want to figure out, hey, what can I change about it to make it more stable and or more active? And so, for example, if I have a glycine here and I say, what happens if I change it to alanine? So mutagenesis and directed evolution are beneficial for this, but the issue is that it can be very time consuming. So if I change this glycines to an alanine, I don't know for sure, right, if I'm going to increase function or completely lose function. But the biggest thing I want to focus on for my talk is that I can't control changes to Km and Kcat. And I feel like this is these are terms that if you're currently in school that are super foreign because it's not something that you really look at anymore after undergrad. But because it's the basis for my talk, I will definitely come back to this. Okay, so recap, we want to increase efficiency and stability. The main way of doing this is mutagenesis, so changing this glycine here for an alanine, or directed evolution, which is completely evolving a different enzyme. Very, very time consuming. And again, you can't really predict what's going to happen if you're going to get a better enzyme or not. The second limitation is that enzymes have short half-lives and in the body. So I gave the example of lactate. Luckily, we produce lactase. But what if the lactase that we are actually delivering is not a human lactase? In that case, it's going to be very tough to keep it stable in our bodies. And so again, enzymes have short half-lives in vivo. So we want to increase the stability. And then lastly, delivery is tough. It's difficult to deliver to certain sites. You have to have the right carrier. And then let's say you want to deliver an enzyme to the brain. Our brain is very, very fragile. Our brain is very, very important. And so we have a blood-brain barrier. There's a barrier that prevents things from getting in that can maybe toxic. So delivering enzymes is very, very difficult. But I really want to bring back this idea of we can't control changes to Km and Kcat. And I want to bring that back because if you can control your substrate binding moiety, which is Km, separate from the catalytic turnover, which is Kcat, you have a lot of control over whether or not you increase your enzymes activity here. So if you have your enzyme and you have a substrate, at what concentration can you get the best binding, or at how low can this enzyme recognize your substrate is going to be your Km and concentration? And then how quickly can we turn that over is going to be into our new product, is going to be our Kcat. And so that is kind of what we're going to look at today. But let's understand a little bit deeper. So again, Km is substrate binding. This is the concentration at which half of your enzyme binding sites are bound with substrate. So the smaller that number, the higher your affinity. And then for Kcat, this is going to be catalytic turnover. This is the number of times that the reaction happened in a given unit of time. So now this brings me back again, right, to my title, Modular Catalysis. Remember what I just stated. We want to control Km separate from Kcat. Back to my example, when we change that glycine to an alanine, you cannot control whether you're changing Kcat or Km, okay? And so that's the modular part of this. What we're looking at is controlling one part of enzyme kinetics from the other, and then exploring Aptamer enhancement of enzyme kinetics in a nanoparticle reactor. I just briefly told you guys what an aptamer is, and we're going to use this aptamer to enhance our enzyme kinetics modularly, okay? So we want to enhance it by only controlling Km versus Kcat, and we're going to do this in a nanoparticle reactor. So what do I mean by this? It has been shown that encapsulating enzymes does increase stability. So there's a big check mark there because I did bring up that stability is an issue. However, the issue that we run into here is that your particles are moving in and out freely, and there's nothing to keep them inside of that nanoparticle. In our case, we're going to use virus-like particles, which I'll come back to. And so as a result, you're getting an increase to Km, and you're going to get an increase to Kcat in most cases that we've seen in literature, okay? So again, you don't have much control still over the Km versus the Kcat. So what do we propose? We propose that by using aptamers inside of a virus-like particle or a nanoparticle reactor of any sort with our enzyme, we can then concentrate down our target with the aptamers binding to it. And then depending on the aptamer affinity versus the enzyme's affinity, it'll simply pass it off to the enzyme. And this is going to drop our Km with no change to Kcat because we are not changing the enzyme, but we are concentrating down our target close enough to the enzyme in order for it to get there quicker. And so, of course, whenever you build a system, you want to characterize each individual part. So first, we have our affinity reagent, which is going to be the DNA cocaine aptamer. We chose cocaine as our target for a couple of reasons. So because this is not a super formal science talk and I'm not in my dissertation defense, I can actually be honest about the non-scientific reason behind it. This project started in 2019 and then COVID happened. And so as we were running out of time with funding and whatnot, we kind of had to change our target here. And so we chose a target that already had an aptamer so that we wouldn't have to evolve it. Evolving aptamers can take years at a time. And we also chose a target that had an enzyme already because, as I mentioned before, evolving enzymes can take years. These are whole PhD projects sometimes. Okay. And so we have our DNA cocaine aptamer as our, with cocaine to be our target. And we then have our cocaine hydrolyzing enzyme, cocaine esterase. And together we would explore encapsulating these in virus-like particles. So whenever you have a full system, you want to characterize each individual part to establish your baseline before you put it all together and say that you've made it better than what it was. And so we're going to start with our aptamer. So I did talk about this cocaine aptamer in my last talk, so I won't spend too much time on it. But we have some characterization goals for each individual component, okay? We have some requirements here. One, we want to generate aptamers with a range of binding affinities to cocaine. Why do we want to do this? If you remember, and I will keep on honing in on this point, we want to drive Km down. Km is the affinity of our enzyme. So we want affinities that bracket our enzyme's Km. Literature says 5 micromolar, but of course our conditions are going to be different, as you will see. But if our enzyme Km, let's say hypothetically is 5 micromolar, we want aptamers with a lower Kd, which I will also describe what that is, or a lower affinity than that. And we also want aptamers with a higher affinity, and then also aptamers that have an affinity that matches the enzyme, because we want to see to what degree can we drive our Km down. So if we have an aptamer that binds much better than the enzyme, the cocaine won't ever get to the enzyme, right? So our Km probably won't change much. But if we have an aptamer, let's say, that is much worse than the enzyme, then, okay, it may not keep the cocaine inside of the VLPs efficiently. So again, we want to have a range of binding affinities to cocaine because we want to see to what degree can we control our Km. And then secondly, we want to be sure that our aptamers don't bind our hydrolysis product. Cocaine esterase, in our case, we use cocaine esterase. At the time, we were actually looking into a few different esterases that can hydrolyze cocaine. But because we didn't know at this time which esterase we would use, we looked at the two different hydrolysis products, EME and BE. So basically, cocaine esterase, there's a class of esterases that hydrolyzes into EME and another class that hydrolyzes into BE. And at this point, we didn't know which enzyme we would use. And so we just wanted to be sure that our aptamers didn't bind EME or BE, because once the esterase cleaves or hydrolyzes our cocaine, we're not going to have aptamers that bind EME and BE, and they won't be available to bind cocaine anymore. So we just wanted to be sure that we weren't taking up our aptamer space with the byproduct here. Okay, and so our first objective, of course, is to generate aptamers with a range of binding affinity to our target, which was cocaine. And how we did this was we did not reinvent the wheel. As you remember, I said that we chose a target that already had an aptamer evolved. And all we did was we took that parent aptamer from literature and the mutants from literature. So someone took the parent MNS 4.1 and made their own mutants GC and GT. And all we did was we took a guanosine-like nucleic acid, inosine, or nucleotide inosine, and we changed these G's here that you see highlighted in pink to inosine. We only did that because we wanted to change the range. We wanted a range of affinities, but we didn't want to completely perturb the binding. Inosine simply has one less amine than guanosine, so we should not perturb binding too much with our aptamers because there's not much of a difference in those two nucleotides. After we did this, we used microscale thermophoresis, or MST, which I won't go too much into that technique. And we successfully got this beautiful binding curve here with the midpoint being our KD. Okay, so enzyme Km is our enzyme affinity. It is where half of our enzymes are bound. Aptamer KD is the same thing, our aptamer affinity, but this is where half of our aptamers are bound. And as you can see, we successfully generated a range of binding affinities to cocaine, ranging from about 80 micromolar to about 230 nanomolar. So that was perfect because, again, we think the enzyme Km at this point in time was 5 micromolar. So that means we have 80, which is much worse than 5, and then 230 nanomolar, which is much better than 5. So we had a really good range to work with here. And then if you don't forget, we also wanted to show that our aptamers did not bind to hydrolysis products. Again, at this stage in the game, we did not know which enzyme we would use. And so we just tested it to both EME and BE. And just showing one aptamer here, we saw this trend for all of our aptamers with this with the absence of that sigmoidal curve. As you can see, we do not get any binding to our byproduct, which was beneficial, and that was exactly what we wanted to see. So then once we characterize the aptamer, and I say I want to note this was actually done in parallel. So we worked at Emory on the aptamer portion originally, and then the enzyme portion we worked on together with Georgia Tech, and then Georgia Tech did all of the packaging. So I'll get into that as well. And so we also had some enzyme requirements. I learned really quickly that cocaine esterase is very, very unstable. But one requirement we wanted was less than or equal to 65 kilodaltons. So MG Finn's a great guy at Georgia Tech. The Finn lab has successfully packaged as large as 65 kilodalton enzyme in the MS2 virus-like particle. And so we knew that they were capable of packaging at the most 65 kilodaltons. So we needed an esterase that was going to be greater than or equal to, less than or equal to 65 kilodaltons. We also wanted a Km at 30 degrees Celsius, at least an order of magnitude lower than 80 micromolar, because this was our lowest binding aptamer. And we wanted to have aptamers that bracket that Km, right? And I'm going to tell you right now, we struggled to get anything worse than 80 micromolar without completely obliterating the aptamer binding. So we were kind of stuck at this being our highest point here. And then lastly, can we characterize this using an analytical method, LC or UV, right? Because you can have an enzyme, but enzyme kinetics is not something that's very, very simple. So for example, our original target was acetylcholine, but the problem with acetylcholine is it doesn't absorb. And so how are we going to track, the loss of acetylcholine? So with cocaine, luckily it does absorb. So we wanted to just make sure that we could characterize this using an analytical method. And so the first objective was to express the unpackaged wild-type cocaine esterase and determine kinetic parameters. Why did we want to do this? We're arguing that we can enhance enzyme efficiency by packaging it with aptamers. So we have to establish a baseline, right, with our unpackaged enzyme. This is going to be our control going forward. This is what we're comparing everything to. So just briefly, I want to go through the steps that I took. I made serial dilutions of cocaine. I want to note cocaine is not brown. Unfortunately, I just could not show a better serial dilution in Bioremediation with clear because it is a clear solution. I then took this cocaine and I added to an 8-cell cuvette. It's important that these are all together because it's quick, right? You don't want it to start hydrolyzing before you add your enzyme. So if I add enzyme to this one, and then by the time I get to this one, this reaction has already started. So I use an 8-cell cuvette with a fun multi-channel pipette. And I then read the absorbance at 240 nanometers because this is where cocaine absorbs. And all we did was we tracked the loss of this cocaine concentration over time. And I generated a very beautiful Michaelis-Menten curve from this. I was very proud of it. It was so beautiful, I actually almost put my name on it as well, Michaelis-Menten Manuel. But even though we have these kinetic parameters, what I really want to pay attention to just for now is this Km here of 10 micromolar, because now we know which aptamers we need to use, right? And so we fully purified it and we were able to successfully characterize it by UV, which I was really excited about. And so lastly, we now get to the fun part. We did all of this work for about 3 years. And finally, in my last year at Emory, we finally had these co-encapsulated virus-like particles. And we were like, okay, let's see what it looks like now. And so I want to note really quickly, a virus-like particle is just the virus shell. you take a virus. In this case, we took a bacteriophage. So a bacteriophage is a virus-like agent that instead of infecting humans, for the most part, it infects bacteria. So it's like a virus to bacteria. Viruses infect us, bacteriophages infect bacteria, in a very, very similar mechanism. And so all we did was you just take the virus, in our case, MS2, and you break it open, remove all of the proteins, all of the nucleic acids, and reassemble it, and just use that outside part as a shell. And that shell just becomes a carrier for whatever you put inside of it. And so some of our characterizations, much like our aptamers and free enzyme, we wanted our VLPs to reassemble around all of our cargo. So if you remember, we knew that he could do a 65 kilodalton enzyme, which our cocaine esterase was actually 68 with the his tag. We knew that was successful, but what happens whenever you're trying to package the enzyme plus aptamers, right? We didn't know how this would go over with an enzyme that size and trying to get some aptamers in there as well. Secondly, can this be characterized using an analytical method as well? So will the VLPs and aptamers absorb at 240? Would the absorbers at 240 also interfere? Think about it, right? VLPs are made up of, is from proteins. And so proteins absorb at 280, aptamers and nucleic acids absorb at 260. These are just their max peaks. You still get some absorbance at 240. So will this be too much interference? Will we be able to get rid of that noise? And then lastly, hopefully we see a decrease in Km and no change to Kcat. And that was the big question there, our big hypothesis. And so first we had to, of course, package our enzymes and aptamers and VLPs. Our objective was to package cocaine esterase into VLPs with and without aptamers. While we're comparing everything to free enzyme, we also have to remember that we need that control, right, of enzyme inside of VLP, no aptamer. And so just briefly, how did we do this? Dr. Shulman Das in MG Finn's lab did a fantastic job developing this brand new method for this. But what he did was he took this MS2, the bacteriophage virus, and he disassembled it using acid, super simple. Excuse me. And then he slowly reassembled just using base in the presence of our enzyme here, which is our cocaine esterase with a his tag binding aptamer, which I'll explain in a second, with or without our cocaine aptamer. Now, what I want to note is that this capsid protein here, the shell of the MS2, is going to be positively charged. Okay, so lucky us, cocaine aptamers, DNA, RNA, they both have negative backbones. So there's going to be a natural attraction to want to bind the inside of the capsid and then in that way form inside of the VLPs, because these VLPs just randomly assemble whatever's in solution. Unfortunately, our his tag in these conditions is going to be positive as well. And we know positive capsid and positive his tag is going to run a repel. So Shulman had the fantastic idea of taking this his tag binding aptamer, so a separate aptamer, that's going to specifically bind our his tag and be negatively charged. And that negatively charged aptamer is going to drag that enzyme to the inside of the capsid, because the inside of the capsid is going to be positive. Fantastic work by Shulman. He was also able to control how many enzymes he got into the VLPs, which was at least one, no more than two, because you also don't want to add that in as well, where you're having two enzymes inside of VLPs, and then that's another component to kind of consider. And if you remember, I told you guys that our enzyme Km was 10. If you remember, we thought it was going to be 5, but we didn't know which aptamers we were going to use to co-package. We just know that we want to bracket 10, a Km of 10 micromolar. And so we chose 80, which is going to be our low affinity aptamer, so a much worse affinity at 80 micromolar than our 10 micromolar. We chose our GC here, which was at about 15 micromolar, so that's going to be comparable to our 10 micromolar Km. And then we chose 230 nanomolar, which is going to be much better than, or much more competitive than our Km. But then I realized a big mistake, and I really want to talk about this because I'm happy to say in my 4th year, I was still making these types of mistakes. Way back when, so the aptamers that I showed you guys, we published that paper almost a year before we were wrapping up this project. And when we published that paper, we were characterizing our aptamers at room temp, but our enzyme kinetics is going to be at 30 degrees Celsius. And so I had to backtrack with maybe 2 weeks left to finish this because my advisor had left Emory or was leaving Emory. And I had to characterize my aptamers now in my kinetic conditions. And the stress I had and the ways I wanted to cry, because if these aptamers, which were some of them were unstable, if these aptamers did not bind, these three specifically, did not bind with the same affinity. So let's say this went from 80 to 20, right? Now is no longer that super low affinity aptamer compared to our enzyme anymore. So I had already done my enzyme kinetics. And so I went back and I characterized these three aptamers again. And luckily, they all changed barely. So we went from 80 to 98 micromolar, which was actually better because it was great to compare 98 to 10 versus 80 to 10. Our comparable aptamer bound went from 15 to 8 micromolar, which was even closer to 10, which was great. And then this one barely changed. We went from 230 nanomolar to 330 nanomolar. So it was still much better than our enzyme. And so the bracketed aptamer still bound cocaine at 30 degrees C, and they didn't change much. So I was really, really happy about that. And I definitely went home and breathed a sigh of relief. So briefly to recap before we get into the answer of our question. One, we've chosen a method for kinetic analysis. We've chosen aptamers that bracket our enzymes affinity. So just remember our enzymes Km, which I want to be very clear. Km and Kd are different. This is a kinetic parameter. So I want to be clear. This is when half of our enzyme molecules are saturated. The Kd of an aptamer is when they're bound. And so I just don't want that to get confused. But our enzyme Km or our enzyme affinity is going to be 10 micromolar, with our aptamers being 98, which is much worse than 10, 8, which is comparable to 10, and then 330 nanomolar, which is going to be much better than 10. And then lastly, we've confirmed VLP assembly and efficient enzyme and aptamer packaging. So we were on a roll at this point. Now, don't forget, this is about 4 years worth of work, though. Okay, so then lastly, right, now we want to package cocaine aspirations of VLPs with and without aptamers. What I want to note going forward, we will use P for package and U for unpackage. And the number that you see with it is how many aptamers on average is packaged inside of the VLPs. So P0 is going to be just our enzyme alone. P3 to 5 has 3 to 5 aptamers. P9 to 10 has 9 to 10 aptamers. P21 to 22 has 21 to 22 aptamers. Okay, so if you remember, we're trying to drive Km down. So not only are we comparing aptamer affinity, we're comparing aptamer affinity and an amount of aptamer in there. Okay. And so finally, the first question we had was, well, what effect does an aptamer with a much lower affinity than our enzyme have on catalysis? Okay. And this is a lot, and I promise I will break this down. But just a brief, again, P0 is going to be package 0. P3 is package with 3 aptamers, P8, P22, and so forth. Kcat over Km is going to be our enzyme efficiency, okay? And so what we see here is that as we increase our number of aptamers, we also slightly increase our enzyme efficiency, but mainly at P8, excuse me. So when we have 8 aptamers with a very low affinity aptamer, we see our enzyme efficiency increase when we have 8 aptamers. And if you look here as well, we do get a significant increase. We also notice that once we hit 22, this decreases. And I'm actually not very surprised by that because now you have so many aptamers that you're likely just outcompeting your enzyme. But remember what I wanted to say. We want to increase efficiency. So we do want this number to go up, but we want this to be strictly dependent on Km. How do we know that this is due to the aptamers? We see that after P0, our enzyme Km goes down, right? And of course, it goes back up at P22. We're hypothesizing here that this is going down because we are now sequestering that cocaine in close proximity to our enzyme. And then successfully, as you can see, we do not get much change to our Kcat here. So let's observe that. What do I think is going on here? Why does P at 22 have no effect on catalysis? I think, again, whenever you have 22 aptamers in there, you are simply going to have too many aptamers that won't be able to really pass it off to the enzyme. So you're outcompeting. So we see our Km go down a bit just in general, and we see no change to Kcat. So then we ask, okay, well, we see what happens whenever we have a much worse affinity aptamer. But what effect does an aptamer with a similar affinity have on catalysis? And we see the same trend here, except we increase our efficiency much quicker. So once we have 5 aptamers, you can see that our efficiency starts to increase significantly. And then it increases even more once we get to 12. And then, of course, we go back down to where we were once we get to 21 aptamers. I'm not surprised by this trend as well. And again, we can see that this is also Km dependent. So we see our Km decrease, and then it goes right back to normal once we get to P21, because again, at P21, now the aptamers are just hogging the cocaine. You have 21 aptamers versus 1 enzyme. And then lastly, we see that we get no drastic change from our unpackaged at the Kcat. So what do we think is happening here? As you can see, we hypothesize that you're just getting an equal sharing between the aptamer and the enzyme because their KMs, or excuse me, their affinities are similar. And then lastly, we want to know, well, what effect does an aptamer with a much better or much higher affinity have on catalysis? And what we saw is that we got no change really to our Kcat or Km, and actually we got a little bit of a decrease, a significant decrease there, which also was not very surprising. If you have a much higher affinity aptamer, the enzyme will never be able to access that cocaine. Okay, and as you can see, we get no significant change to our Km here either, and our Kcat stays relatively the same. And so again, we hypothesize that that higher affinity just simply outcompetes our enzyme for the substrate. Okay. And so we got no change to Km, no change to Kcat, and it's just simply that we believe that the aptamer is hogging the cocaine there. So in conclusion, we are, to the best of my knowledge, I got in trouble for not saying to the best of my knowledge. So to the best of my knowledge, we're actually the first to show that co-packaging of aptamers with enzymes gave a Km-dependent increase in catalytic efficiency. These studies provide some clues about strategies to improve performance further. So we only got about like a 2.5-fold increase, which isn't super attractive in therapeutics. What the most attractive part is that we were able to do this Km dependent as much as possible. But it would be really great if people could try different encapsulants, right? And different affinity reagents and different maybe types of aptamers that could increase their performance even better while still keeping that Km dependence. This suggests that faster off rates may be an important factor that we have yet to explore explicitly. So off rates meaning when the aptamer releases the cocaine. And an added bonus, again, VLPs have been shown to protect enzymes from degradation and enhance delivery. And so one big issue that people were running into is, okay, we can encapsulate it, great, that we protect our enzyme. We can choose our encapsulant, great, we can enhance delivery to certain spots such as the brain. So you can functionalize VLPs with receptor binding moieties where it can bind the receptors to get into the brain, right? So those things were already sorted out. But one big issue people run into is once you package these enzymes, sometimes your efficiency will decrease. And so one thing we answered is that we can increase efficiency, and we showed this this component of control as well. And so with that, I would actually like to acknowledge my previous lab and my previous funding sources, especially for funding this project. I had a great time working with these guys. And I want to give a special shout out to the Fin Lab at Georgia Tech. I learned a lot from that lab. I thank them a lot for allowing me to come and learn how to package my cargo into VLPs as well. And with that, I will take any questions.
Fantastic talk. That's super cool work, and you did a great job explaining it. So we'll just go ahead and jump straight into the questions here. First question is, what kind of science are you doing now, and does your grad school work tie into that?
Great question. So now I am in structural biology, which is, every time I say I work for Mike Summers, people are like, the NMR guy? And so I guess briefly, he studies HIV genome packaging.
Mm-hmm.
He studies viral genome packaging in general, but he really focuses on retroviruses, especially HIV. And one big find, so humans, we cap our mRNA in the transcription, or it's post-transcriptional, but in that cellular machinery, we cap our mRNA to prevent being cleaved and degraded. And HIV over time and other viruses over time have come to adapt that same mechanism in order to survive and hijack our system.
Okay.
But what Mike's lab found is that people always thought that capping of that 5' end was only for translation and transcription. But what he found is that it's also for packaging. It's very, very important for packaging. And so I am now studying that in MLV, which is a mouse virus. I'm studying how that capping affects packaging and different types of retroviruses.
Okay.
So yeah, and I'm using NMR to do that as well.
Okay, that sounds super interesting as well. Maybe we'll get you back a third time. That'd be great.
Yes, definitely.
Let's see. You mentioned you never got to work with Aptazymes. What would you have wanted to do with them, and could you use them for work similar to what you presented?
Great question. Yes. So the original project, which I'll go back to the beginning, but this original project was actually we were trying to so acetylcholinesterase is an enzyme that hydrolyzes acetylcholine in our bodies.
Mm-hmm.
And when organophosphates are present, which is typically in your agricultural pesticides, is inhibited. And so what we were trying to do was evolve an acetylcholine binding aptamer and an acetylcholine hydrolyzing DNA enzyme,
Mm-hmm.
and then package those together. DNA enzymes have terrible efficiency, which is why we were actually studying increasing efficiency. And so
Oh,
I would love to see that. I would love to see an a cocaine binding aptamer and cocaine hydrolyzing DNA enzyme together and see if that in itself actually without packaging can increase efficiency. And I would also love to see how that would function together inside of here. So when I say we've opened the door for this idea of, hey, we can control KM and we can increase efficiency, I would love to see what would happen with an Aptazyme packaged inside of VLPs. That would be really interesting. It's a different kind of closeness, right? Because
right,
they're bound at that point together. The aptamer and DNA enzyme are bound. These are still kind of free flowing inside of the VLPs.
right. Yeah, that's interesting.
Yes.
Let's see. Is there an advantage to using a virus-like particle as opposed to something like a reverse micelle?
Great question. I'm not familiar with reverse micelles. If you can briefly give me like a quick,
So the ones that I'm familiar with, you would have like an organic solvent, and then you have like water, and you use some kind of like basically like a surfactant, right? And so
Mhm.
you set up the ratio of the water to the organic solvent, and you have and however much of the surfactant, and you end up with like I mean it's similar to what the virus like particles are, where you have a confined space, and then if you have something uh like a reactant or something like that's water soluble but not soluble in the organic phase, so you basically trap them inside those micelles, and you can control the ratio and the size and all that.
Oh yeah, great question. That's the one question I haven't gotten. I always get asked why not, and they insert whatever carrier.
Uh-huh.
May I ask you this? Can you control the size of those micelles?
To at least some extent, yeah.
Yeah, so I do know with the VLPs,
Mm-hmm.
there are a few benefits. One, you can choose your virus, like the virus core,
Mm-hmm.
to kind of target a specific area in the body. And that has always been very,
uh
very attractive. Yeah, so
yeah.
if you choose a virus that wants to naturally, for example, infect, an intestine, and you want to deliver something to the intestine, you could choose a VLP that has a native, from a native virus
Yeah,
that wants to
that makes sense, yeah.
accumulate there. That was one. And then also you have a bunch of sizes to pick from.
Mm-hmm.
Um, and then of course, in this case of MS2, not all VLPs operate the way that MS2 does, but of course the random assembly, you don't have to functionalize your enzyme or your aptamer in order for it to be
Right.
inside it.
Okay, that makes a lot of sense, yeah. Well, yeah, you're in the biocompatibility, not just
Yeah.
compatibility, but like being able to target something, that's a really good point.
Yes.
That is I can see where that would be a huge advantage.
Definitely, yeah.
Let's see. If you got to continue this work, what would have been the next steps? And in other words, what would you like to see come out of this work?
Great question. I would want to increase efficiency more. So another reason we chose cocaine esterase is because it's really promising
Mm-hmm.
in therapeutics, but it's just so unstable. But the problem is, again, once you package it, then the activity just decreases, it diminishes. And so that's why another reason that it was attractive to use it. I would love to see this in mouse studies. I would love to see this in mouse studies. If I had more time, I would want to increase the efficiency more. So play with more aptamer ratios, more aptamer affinities to see if we could get twofold increase. And if we could, I would love to see how this functioned in a living system for sure.
Yeah. So I guess off of that, so do you envision these as like a treatment? Like if somebody like would this be like an overdose treatment, or like what? It sounds like you have an idea of what an application might look like.
Yeah, I could see it as a promising overdose treatment. We did not intend for that.
Okay.
I don't know I know that they were working on getting some more funding for stuff like this, but I'm not sure what the next steps were for them. I do know that we finished out the project that we wrote in that grant, but I don't know that it's promising enough yet. But I will say, because cocaine esterase is the most efficient esterase. But so packaging it does diminish the activity a bit, but not with the Aptamers, of course, but I would love to see this therapeutic. However, that was not the goal. The biggest goal was is a proof of concept.
Sure.
Can we control Km separate from Kcat? Because no one's, been able to do that.
Right. Yeah. No, I mean, yeah. The methodology here is really interesting. Like, is somebody working on like maybe taking the cocaine esterase and making it more active just in and of itself? Is there any work like that going on that you're aware of?
They target stability more than activity because it is very efficient,
Okay.
but it's unstable. I think the half-life is like 14 minutes.
Oh wow. Okay.
In the body. So it's really unstable. So they always target stability, but then when they do that, it diminishes the activity, which is another big. That's why we wanted to package the native enzyme. We don't have to mutate it, right? We could just package it.
That makes a lot of sense. Okay. Let's see. How do you control the number of aptamers inside the VLPs?
Great question. Yes. So, let's see.
Oh, we got the backup slides.
I had to.
Hey, that's smart. That's smart.
Yeah. So, Showman did a fantastic job optimizing this, but we have a FAM on the Aptamer so that we can, of course, visualize it inside of the VLP. Or not actually visualize it, but you know what I mean? Like when we do our LC, we can visualize it there and quantify it.
Okay.
But it's just a ratio. He just optimized the ratio a good bit.
Oh, okay. So like when you're when he's putting everything together, he knows like if I put, X amount of this part in with Y amount of this other part, and then and everything mixes together and is allowed to react, you're going to end up with an average of.
Exactly.
Okay.
Yep.
That makes sense. Okay, so I guess along the same line. So this was from slide 24, where you're talking about the Kcat to Km ratio. Could you further fine tune the number of aptamers to try and maximize that ratio?
Yes, and that's why I said if we had more time, that's what I would. If we had more time, I would want to try all 8 of the aptamers actually. All 8 that we came up with. Oh wait, where did it go? I would want to try all 8 of them.
Okay.
I don't care how close they are, like in affinity.
Well, yeah,
I don't care if
you never
they're
know.
2 apart.
Yeah.
Yeah, I want to try them all. If we had more time, for sure.
I can't blame you on that one. I mean, saw a pretty clear difference, in the sort of wide range. It'd be neat to see, when they're a little bit closer together. Do you start to, are there any like specific ones that work out a little bit better than the others?
Right, because stability is also going to be a thing too at play. So some of these aptamers are more stable than others. And I don't know. I know that VLPs are. I actually forgot to mention this in the background, but I know that VLPs do protect against protease and nuclease degradation because they can't fit into the little pores.
Okay.
But I don't know about the temperature part. I don't know if it can. I don't know if it can protect aptamers in a temperature dependent way, and so I wonder how, even if it's just slightly less stable at 30 in those VLPs, even that may be an advantage and maybe a disadvantage. So I would want to try all 8 of them.
That's a good reason.
Yeah.
I guess if anybody else has any questions, get them in. I'll ask one other one just out of my curiosity here. So when you swapped out when you swapped in inosine for the guanosine, did you expect it to decrease your KD, or did you have a feeling for which way it was going to shift?
I didn't know like what specifically. So I will admit I was a rising second year once we got to this part of the project.
Mm-hmm.
So at this point, I was actually still trying. I couldn't even tell you. I just remember thinking, why are we doing this? Like,
Yeah,
why
yeah.
are we using Enzyme? It took some time before I got to this point in understanding why we did it.
Okay.
But no, I didn't know. I really didn't know which way it would sway it because I knew one thing I did know was going to be stability dependent.
Okay.
And then also proximity to the active site or the binding site, which was the binding site's here. I didn't want to get too close,
Mm-hmm.
right? Completely perturb it, but I didn't really know. I's want to bind C's just as much as G's do. They
Okay.
also bind A too, but it's not going to be much different than guanosine. So I truly and honestly did not know.
Okay. I was just
Yeah.
curious. That's very cool. So I think that's all the questions we have. So I will say thank you again. Really great talk. Like I said, super interesting work.
Thank
Well,
you.
well explained in a great story, and I appreciate you sharing it with us.
Thank you so much. Thank you so much for having me
Yeah,
too.
absolutely. Anytime. Like I said, whenever your next project is ready for a talk, come on. Let me know. Come on back on.
That's gonna be a cool one. Let me tell you, Ben, just briefly.
Yeah, please.
I hate to even be this much of a nerd. So real quick, HIV,
Hmm.
um, the start site for transcription, so just like the one position on the 5' end,
Hmm.
can have either 1G, 2Gs, or 3Gs.
Okay.
And literally just based off of that, 1Gs are packaged, 2 and 3 are translated into proteins.
Hmm.
With MLB, it only has 1G. And so what I'm studying is how do we choose? How does it choose what's packaged and what's translated? So I will
Yeah.
definitely once we're done with that,
Yeah.
I would love to
Share.
give that
Share,
talk.
please. That sounds really cool. Oh, I will look forward to that
Oh,
then. That's amazing. Yeah. Then
you can go out.
so thank you again for a great talk. Thank you to everybody for coming out and asking great questions.
Can
And
you hear me? I'm sorry,
oh yeah, yeah.
yeah.
And we will be back. I think not for a couple of weeks unless I can get somebody else talking, but we'll be back and we'll have some more great talks. So follow on Twitter at Meekins Lab and follow Brea as well at Brea the Scientist on Twitter as well. And we'll post this talk up a little bit later tonight. And yeah, thank you again. Thanks everybody for coming out and see you next time.